this episode of La Segunda Derivada, the podcast of the School of Engineering of UC Chile begins a season with guests who will talk with Agustín Covarrubias about the current and future challenges of our society. A podcast that will address the role of science and technology in solving the most urgent problems, both locally and globally.
Welcome everyone to a new episode of The Second Derivative, the official podcast of the UC Chile School of Engineering. In this episode, we'll repeat something we did some episodes ago by having a podcast in English. This time, we'll interview Andrea Ravasio, assistant professor in the Biological and Medical Engineering Institute here at the School of Engineering. Dr. Ravasio is currently the director of the Laboratory for Mechanobiology of Transforming Systems and Mechanobiology of Development project. He also holds a PhD in Human Physiology from the University of Innsbruck and a Master in Biology from the University of Milan. He has worked in leading research institutes in Italy, Austria, and Singapore, including recent work as part of the Singapore-MIT Alliance for Research and Technology. Andrea's research is mostly focused on mechanobiology, a field that lies in the intersection between biology, physics, and engineering, trying to understand the complex interactions between physical forces and cells. This has important implications not only to understanding fundamental processes in biology, but also diseases such as cancer, and he also teaches an innovative course on the mechanobiology of diseases. Welcome, Professor.
Thank you very much for having me. I look forward to this conversation.
I look forward to it too. So, just to get started, one of the first things I noticed about your profile is that you're a biologist by training, working in the cutting edge of engineering, physics, and medicine. How did you get into these lines of research? What caught your eye about mechanobiology?
Mechanobiology wasn't even a discipline when I studied. It is very recent, maybe the last 15, 20 years at most, and unfortunately I'm older than that. So when I started, when I had to finishing my secondary school, I didn't know what to choose, physics or biology. So what I did, I just took a train with my brother and went for a travel around Europe. And when I came back, I decided to go for what I thought was more difficult, so biology. But I always had an interest in the physical aspects of biology. So I kind of self-taught myself during the undergraduate study as much physics as I could. And then when I did my master and PhD, then I chose physiology, which is the most physical of the biological sciences. And then during the PhD, I actually did it in a lab that was working on biophysics, particularly lung biophysics and membrane biophysics.
So it looks like you always had a knack for physics, including physics in your biology studies.
And biology.
That's really interesting.
And then it came also the interest for medicine, because the lab where I was working, the biophysical lab where I did my PhD, was in the faculty was a medical university. So we were always trying to find ways to apply what we did for the larger benefit of the society, like developing assays. So I developed an assay during my undergraduate, like a side project to screen for surfactant function, which is what allows people to breathe effectively.
So when you started all of your undergraduate studies, do you think you would arrive at something like this?
Oh, yeah, it was my childhood dream. Yes.
Was it really?
Yeah.
Not to say like...
So when I started, when I was a kid, I always dreamed of being a scientist. So for me, it's not even a job, this one. It's just something I do with passion.
And did you imagine you will travel the world doing it?
It was my other passion, because I'm Italian, but originally I was born in Africa. So I always had it in me to travel as much as possible. And yes, so that made me some kind of scientific gypsy. I never settled down anywhere, but just try to find the next challenge.
I love that.
Cheers.
I think maybe the scientific spirit has much alike with travelers in general.
Yeah, I agree, yeah.
Looking for the next frontier.
Yes. It may be in science, it may be in the actual world, but yes, we're always looking for the next frontier.
So moving on to your research more generally, transdisciplinary fields are often hard to define, but what do you think are the core questions or approaches that underlie mechanobiology?
Yeah, I don't know. There is this famous story. I think it's an Indian tale where it tells of these many people that are blindfolded, and each one of them is touching a different part of an elephant. And the one that is touching the trunk thinks is a tree. The one that is touching the tail thinks that is a sweep, and so on. And the one that is next to the ear thinks that is a fan. So this is telling that one unique approach is never sufficient to understand the complexity of a system like the biology is. So that's why I always like to work at the interface between the different disciplines. Also, another motivating factor for me is not being limited, but what are the tools that we have at hand. So, just like to give you a little bit of an historic view on biology. So, it started off mostly like a branch of chemistry. So, the type of technology that were developed for chemistry have been applied to study biological systems, and this has continued until very recent. And biophysics has always been some kind of a unique branch in the biology world. So this has been very limiting because at the moment what the type of discoveries that we are making are mostly based on the chemistry of a biological system. So the cell is often seen like a chemical factory, and all the mechanics, all the important physical cues that the cell has to, which are important for a number of reasons. Maybe we'll go into more of my research on development and cancer later, but they're very important for cellular function, and they actually determine what cells do. So the genetic code is determine what the cell is. But then the mechanics, the ability of the cell to interact with this physical world determines what the cells can actually do in practice.
So it's interesting because thinking historically, the convergence between chemistry and biology was an important turning point for biology. It changed the way we looked at cells and living organisms, and it allowed us to use the tools of science to understand the lower level mechanisms that make cells work. In the same way, I think physics, as a more recent joining approach to understanding biology, seems to have been...
Physics and engineering.
Physics and engineering seems to be building this new turning point for biology, for understanding aspects that we may have not considered before.
Correct, yeah. No, that's exactly the point. So in my lab, what we do develop our own methods to study the biological question that we have. So in that sense, we are not limited by the tools because we make our own tools. And one of the cutting edge technologies that we use is microfabrication, which originally was developed for computer science to build transistors and processors. We use the same type of technology to build microfabricated devices. They are micro, so they're in the same length scale of one cell or one molecule. And so using those one, we can actually interrogate how this unit work as functional unit that develop the biological world, basically.
So microfabrication, as I understand it, is this very precise manufacturing process that allows to build devices or microarchitectures.
Correct.
So in a way...
For instance, I can give you just a very easy example. We can build a microscale spring that deforms when the cells are pulling on it. So we can measure the forces exerted by one cell, like a myocardial cells. So this has been been implemented by some colleagues in US. Christopher Chen was a pioneer there to develop high throughput screening for drugs that can effectively cure some diseases in heart defects, for instance.
That's really interesting. So do you think maybe the emergence of these tools have opened new frontiers of research? And was that recent when these kinds of tools appeared?
That has been pioneered. That has been pioneered by Professor Whitesides in the MIT and Harvard. And he was a material scientist working on the development of these type of technologies for engineering approaches of engineering technologies. And then he discovered that one of the material that were commonly used as an insulant is called PDMS, polydimethylsiloxane, is also biocompatible. So then he started to use these microfabricated devices as a mold to then mold cast these type of devices on which then you can grow cells.
So biocompatible. So you're talking about taking this material and place it in an organism.
Yeah, you can effectively. So this the most widely used application for this PDMS in the real world case scenario is for contact lenses, because it's not only biocompatible but it's also completely transparent to oxygen. So it just allows. Before the contact lenses were also made of glass, which you can imagine you have just a glass attached to the eye.
Yeah, that sounds actually dangerous.
It is dangerous, and also it's the glass has no permeability, so the cells were dying because they were asphyxiating them. Instead, now they make them of this soft gel and perfectly transparent and biocompatible.
I never thought about it, but while hearing you talk about it, yeah, I mean, when you make contact lenses, you probably need to worry about how the cells underneath are actually working, and if there's no permeability, there's no interface to the environment, they'll probably start to die.
Yes, correct. And yes, so other type of applications. So one of the things that we are most excited about is the creation of biomimetic environments. Why that? So at the moment, there is this dichotomy between the basic science and the clinical applied science. Basic science is run on biological model like cells or insects or embryos extracted from frogs or whatever other animal. And instead, clinical research is done on animals, rats, mice, going up to also monkeys. What we do, we bridge these two worlds, and so we create in vitro systems that we can manipulate in the lab, where we can grow cells and make them behave like as if they were in the real body. So on one way, we also solve the ethical problem of intensive animal usage and sometimes unethical usage of animal models. And on the other hand, we provide a system that, because it mimics the in vivo conditions, but it can be used in laboratory settings. We provide a system where we can experiment on the cells and have the accuracy of visualizing the cells with the high precision tools that we have nowadays, like super resolution microscopes, where we can look at the forces of the cell. We can look at the localization of the molecule within a cell with the accuracy of nanometers, which is unprecedented.
So it seems like a good part of your research is not just inquiry by itself, but also developing novel approaches to allow you to just begin to start the inquiry. So is that really part of the work? Do you spend a good part in engineering the research, so to speak?
Yeah. No, absolutely. So my lab, we do three things. The first one is investigate basic science, so to understand the principle by which biological system works. The second, which is very important, is development of this type of technologies. And the third, which is what we are entering now, is to develop screening systems or diagnostic tools or manufacturing tools to create the next generation type of therapies, which take into account also what is the mechanic of the system. Like I said before, so at the moment I'm writing a grant on CAR T cells. CAR T cells are the frontier to cure cancer. So you take T cells, which are cells from the body of the patient, which naturally are trained to kill cancer cells. They just don't do it very effectively. So what has been done in the past year was to increase their efficiency by transfecting it, by putting a molecule inside the cells that makes them to recognize the cancer cells better. But the problem of that is that sometimes they overshoot. So at the moment there is only a handful of therapies that are approved based on these technologies, and they are all targeting blood cancers, and they can be used only in young, relatively healthy people. So people that besides having a tumor, then they don't have other major complications. Why? Because if you put these cells and they're overactive, then they cause this type of cytokine storms. Like most of the patients of COVID were dying because of cytokine storms. So it's an overreaction of the immune system. And so our project, what we try to do is to increase the mechanical specificity of the recognition of the effector cells towards the target cells, so the cancer cell. Why that? Because it appears that when you put these molecules inside these cells, then the cells tend to lose their ability to mechanically interact with the cancer cell. So they're just interacting with the cancer cell in a chemical way. And so our idea is to try to recover the mechanical recognition of the target cells.
So this is really interesting. If I understood it correctly, cancer cells, T cells, are naturally fight cancer cells, and some treatment approaches have tried to enhance the ability of the T cell to actively combat cancer cells. But the problem is they rely on chemical signaling alone. And you have found that mechanical processes that govern the interactions between the T cells and the cancer cells are actually really important to making the treatment more safe and effective.
Correct, yes. Because when these T cells recognize a cancer cell or try to recognize a cancer cell, they first engage chemically. So they have surface receptor that interact with surface receptor of the target. And if that is recognized as a cancer cell, then they just put there a lot of molecules that will just make holes in the membrane of the cancer cells, and like this they're gonna kill it. But naturally what they do, they will tend to take all these molecules and aggregate them mechanically in a confined space, and if they manage to do that, then they will exactly interact in that spot. When you overexpress this receptor, which is what CAR T cells are, when you overexpress this receptor, then they are everywhere in the cell, and so then the cell bypasses this aggregation step. Okay, so basically because there are so many, then they can react everywhere. And in that case, then they're losing also the ability to recognize if that is effectively a cancer cell, because there are also a number of downstream steps that the cells need to check to confirm. Is that a real kill, or is that a miss like friendly fire, let's say? And this recognition requires the mechanics.
Wow. So I think that's actually a thing that has happened many times in the course of like medical treatments, in the sense that once certain biological systems are understood better, treatments tend to approach the system in a more holistic manner. And that sounds really abstract and stuff, but when you're talking about pharmaceutical treatments, by example, think about all the different ways cells can interact between each other actually matters a lot, because otherwise we are only emulating part of the behavior and the results are suboptimal, as you've described.
Yeah, perfectly said. I need to put to just give a caveat here, which is that so I'm overly simplifying some of the steps. So for the communication purpose, I'm skipping some of the details on the thing. Of course, things are way more complicated, as normally they are when you approach a system like biological system, and especially a system like cancer, where things are very, very complex. But then another major issue of this type of therapies, which brings back to our technological effort, is that they are very, very expensive, because the manufacturing of these cells, it is intrinsically expensive, because you need to take cells from the body of the patient, expand them using a very, very expensive reagent, and you need to expand them to have millions of cells before being able to re-put them back in the patient after you have increased their efficacy. And so the idea is also to develop manufacturing systems that do not require so much of these expensive reagents, so that you can somehow democratize a little bit more these therapies, bring them to the bedside of every hospital in the world, not only in rich people hospitals.
That's impressive. So how does that actually look like? You talk about the traditional approach, which is taking cells out of the patient, treating them with chemical reagents, and then putting them back. So would this work the same way, or would you alter something?
The overall idea is that if you need fewer cells to do the same type of work, and these cells are also trained to better recognize the actual target, then all the chain will just shrink. Instead of having to expand them to millions, maybe you need only 1,000 or hundreds of cells.
And getting costs and time in the process.
Exactly, yes. Mm-hmm.
So, okay, that really sounds awesome, but I have questions. When you arrive at the lab, how does your research actually look like? What are the kind of things you're doing during the day?
Yes. My research now is exciting on the intellectual point of view, but boring on the practical one. I just look back at the time I was a student or a postdoc, and those were the fun times where I could go to the lab and actually get my hands on the thing, and that was where it was most exciting. Now my days are mostly writing paper, writing grants, answer emails, deal with bureaucracy. So, but yeah, I think that. So at the moment, the most exciting thing is to allow my students to be successful. That is what allow my students to be successful, and how they can be successful is if they start their own company based on the type of technology they can develop, if they publish high impact factor papers. So see them happy makes me also happy. Not sure if you want me to tell you what was an exciting moment of when I was actually working in the lab. But
I'm actually really interested in that.
There are few moments that are these true eureka moments, which are what tricks you in being a scientist. Because overall, science is a resilience job in trying to sustain one failure after the other until you reach this eureka moment, which is I think it is as close as possible to this explorer when they find, I don't know, the Temple of Doom or things like that. And so for me, probably the first eureka moment was we have been running this experiment on lung surfactant when I was a PhD student. We ran this type of experiment over and over and over. But due to technical limitation, we always did this type of experiments at room temperature. Then one day I just decided to spend a little bit of time in developing a little device so that we can put the same system on the microscope like we always did, but under controlled conditions, so I could change the temperature. And I put it on 37 degrees, and instead of seeing this type of nice, complex, two-dimensional structure, I see them emerge in 3 dimensions. And I did not know why, but I was sure that I was the first one to see in the world. and so you had this fulfillment of being a real explorer.
Yeah, I mean that's a common narrative that many scientists share. Some people think about science of being like this really exciting one eureka moment after the other kind of thing, when actually, yeah, it's for the long haul. You have to be really invested in what you do just to have maybe one in a few years eureka moments that.
Exactly. And then you always see your professor like being this guy that knows everything. But the type of student that we mostly appreciate are those ones that we learn from, those ones that bring us their eureka moment. So this is what I was referring before, to see them happy. See them that they are successful, that they work hard and they come up with their own eureka moment, plus the deliverables, which are the patents, the publication and so on. So this is what satisfied my day-to-day work nowadays.
Yeah, I imagine that's really gratifying. I mean, all of this work put into empowering students to continue these kinds of research and taking to a new level and effectively seeing that work. Okay, so in this podcast, we regularly talk about open questions that appear in different disciplines. Maybe some of the stuff that makes scientists curious about what's the frontier, what's unknown. In your opinion, what are the biggest or maybe most interesting mysteries you've come across in your research? What areas are unexplored?
Everything we do is rather new, so I feel quite lucky to have stepped into this line of research because it is fresh, new, out of the box, new discipline. So we are actually rediscovering a lot of old rules, but now we look at them through the magnifying lens of their mechanics. So what was considered to be mostly a biological process, now we rediscover a lot of them to have also a lot of mechanical basis, multiscalar. So what are the frontiers in mechanobiology? I would think that a lot of people are actually investing time in investigating the immune system because of the type of output that we have, cancer. So it's moving towards more the biomedical type of world. So he's coming out of his early year of niche type of cool science, and now he's getting into the real world. So as you see, then what we are actually doing in my lab is in line with what the world of mechanobiology is going towards. So we are developing novel technologies that can be useful for biomedical investigation and also biomedical treatments. And we are developing. We didn't explore a lot there. So what are our basic questions? But I think that is a little bit too specialized for this type of audience. And then we are trying to enter the world where we can actually make an impact for the people that are really in need for novel therapies.
So it was surprising to hear you talk about this mechanistic and physical aspects of cellular biology, saying there's really important interactions that maybe we haven't considered before, and that suddenly have all of these really important implications to the biomedical community, by example. So I took cellular biology off the cell, which is actually an introductory course, of course, but we never heard of physical processes. It was all chemical signaling. So do you see this changing anytime soon?
Yes. So the knowledge of mechanobiology is slowly entering or creeping into the textbooks. So now the Alberts, which is the book where everybody studies cell biology, is taking into account some of the mechanobiology perspective. So yes, I think that soon we'll see more students studying the mechanics of the cells and not only the biochemical regulations of that.
Okay, so I'm extremely interested about all of the things you're working on. It seems like for a field that, is really recent in its emergence, at the very least in the scale of biological history, you're doing some really intense stuff in very short time periods.
Yeah, and I have to say that Chile has been quite gracious to me in this sense because first of all, I'm enjoying a nice country. And second of all, I've been lucky enough to win several grants. So now my lab is fully equipped and we have a lot of resources to actually do cutting edge investigations. So we are competing, or better, we are collaborating with the top groups in the world, big universities. We don't shy down to anybody in terms of our capabilities, what we can do actually here in Chile. I have to say that I also enjoyed a lot being a pioneer. So because coming to the country or in the continent where this field is not developed, mechanobiology now is mostly done in the hotspots. So in Singapore there is a mechanobiology institute, in the MIT, in the big university in Europe, you have people working on mechanobiology. Not many people in South America are working on mechanobiology, and so I could count a handful of people. And definitely we have been pioneering this science in Chile, which brings its advantages.
I'm really excited to know that we're actually working on that front, and we're actually giving it a fighting chance in the international scale, not just the national local scale. I mean, this is stuff that's really on the cutting edge of research, and it's exciting to see it around.
Cheers, thank you.
Well, so that's about it. Thank you really, Andrea, for coming today. I mean, we had a great time, and I'm really interested to keep learning about this stuff. It's definitely not, it definitely hasn't ended for me. So, yes.
Yeah, thanks a lot to you for the invitation. It has been great. I had a lot of fun. Thank you.
And for the audience, keep watch of the next episodes. We'll be releasing a different type of podcast very soon, a more narrative approach to podcasting, not so much interviews. So activate updates on your favorite podcast stuff. Thank you.
Nos vemos en el próximo podcast, donde continuaremos abordando cómo desde la ingeniería podemos aportar al desarrollo de los países.